Hello everyone, this is Olali, and now we'll talk about the topic of osmosis. Osmosis means water diffusion. So here the molecules do not move, but water does. For osmosis to happen, we have to fulfill two important conditions. Number one, the membrane is not permeable to solutes. So those molecules here on this side of the membrane are non-penetrating. They cannot cross the membrane to the other side. The second is water can go from one side to side, and there is a concentration difference between the two sides of the membrane. Since the molecules are non-penetrating, water moves to them. So water moves from the side that has lower molecule concentration to the side that has higher concentration of molecules of solutes. At the end, the concentration is going to be equal on both sides of the membrane. But of course, one side of the membrane has more water than the other. However, again, the concentration is equal. So here, this side has more concentration of solutes. This has less. But at the end of the osmosis process, after water moving to the side that has higher concentration, now the concentration is equal on both sides of the membrane. The unit of concentration is the osmolarity, and water moves through channels that we call aquaporins. They are found in the cell membranes. So what is the unit of the osmolarity? The unit of the osmolarity is osmol, and each osmol has 1,000th milliosmoles. So how to calculate the osmolarity? For example, if we have a compound that like sodium chloride dissociates in water into sodium and chloride. So here you have to multiply by 2. For example, to calculate the osmolarity of 0.15 mole of sodium chloride, to change the mole into osmol, you have to multiply by 2. And then it's going to be 0.3 um, osmoles. And this equal 300 milliosmoles if you multiply this by 1,000. So here we have a membrane, and again the molecules here are not penetrating to that membrane, so osmosis has happened. So here water goes to the side of lower molecule concentration to the side of higher concentration until the concentration is equal on both sides of the membrane. The solution that has lower concentration is called hypotonic solution. The one with higher concentration than the other is hypertonic solution. Okay, what is the difference between osmolarity and tonicity? Osmolarity is the total concentration of everything, regardless being penetrating or non-penetrating. That is osmolarity. But do we care about the total osmolarity when we judge osmosis? No. We care only about the non-penetrating solutes. And in this case, we call that tonicity. Tonicity is the concentration of the non-penetrating solutes only, and they have the osmotic effect. Based on that, we can classify the solutions into osmotic or tonic. Osmotic is the total concentration, and tonic is the concentration only of the non-penetrating molecules. The isotonic solution is a solution that has a 300 milliosmoles per 1 liter concentration. So if the osmolarity is 300 milliosmoles of non-penetrating solutes, we call that isotonic. Iso means the same because the tonicity inside our cells is 300 milliosmoles. How about if the concentration of the solution is more than 300 milliosmoles of non-penetrating solutes? In this case, it is hypertonic. If the solution has a concentration of less than 300 milliosmoles of non-penetrating solutes, it is hypotonic. So tonic here cares about the non-penetrating solutes only. On the other hand, isosmotic is the total concentration. So if the total concentration is 300 milliosmoles of everything, regardless if it is penetrating or non-penetrating, this is isosmotic. Hyperosmotic is the total concentration of more than 300 milliosmoles of anything, regardless. Hypoosmotic is a solution that has less than 300 milliosmoles of anything, regardless penetrating or non-penetrating. But again, to judge water movement, we need to know the tonicity, the tonic, not the osmotic. The tonicity inside the cell is 300 milliosmoles. So what happened to the cell if you put that cell in an isotonic solution? So it has the same tonicity that inside the cell. So here, the cells stays the same. Water will not move in or out. How about if you put your cell in hypotonic solution that has less than 300 milliosmoles of non-penetrating solutes? So in this case, what happened to the movement of water? Water moves in the cell. Remember that always water moves to the higher concentration site. So now water enters the cell would lyse or rupture. If you put these cells in a solution that is hypertonic, meaning has tonicity that is more than the tonicity inside the cell, so it has more than 300 milliosmoles of non-penetrating solutes, this means that water will exit the cell and the cell would shrink. So here, this what happens to a cell. If you put this cell in isotonic solution, the cell stays the same. If you put the cell in hypotonic solution, water enters the cell toward the higher concentration site, and the cell would lyse. And if you put your cell in a hypertonic solution, water leaves the cell out to the hypertonic side, and the cell would shrink. Anything but isotonic can damage our cells. That's why our plasma around our body cells must be isotonic, must have the same tonicity as inside of our body cells, which is on average 300 milliosmoles. Okay, so the normal tonicity of the plasma is 300 milliosmol or 0.3 osmol, and the tonicity inside our cells are the same. Now, how to increase the osmolarity or the concentration of a solution? If you want to increase the concentration of sugar in your cup of tea, there are two choices. Either to add sugar, you add some solutes, Or you can take away water. Like if you boil that tea, the water evaporates. So if you take away water, now also this solution becomes more concentrated. So add solutes or remove water. Take away water. How about if you want to make your solution dilated, like decrease its osmolarity? So you can add water. It's going to be diluted, or you can take away solutes, and the body can take away solutes or add water. And in this case, you will make the solution of less osmolarity. So what happens to our blood osmolarity in case of dehydration? You are taking away water, so the osmolarity in this case increases. Okay, now let's go ahead and practice some examples. So what is the osmolarity of water? Water without anything. It has a zero thing, so zero osmolarity. And this is hypoosmotic since it has less than 300 milliosmoles of everything. How about the tonicity? It is hypotonic. So if you put these cells in water, what would happen? Water enters the cell and the cell would lyse, swell and lyse. How about if you have 0.05 molar of sodium chloride and the sodium chloride is non-penetrating? So to calculate the osmolarity, you need to multiply by 2. So that's 0.01 osmol or 100 milliosmol. 100 milliosmol is less than 300 milliosmol. So this is hypoosmotic. And now the solution has less than 300 milliosmoles of non-penetrating. So to classify its tonicity, it is hypotonic. And what would happen to the cell? It would swell and lyse. How about if you have a solution of 0.15 molar of sodium chloride? To calculate the osmolarity, you multiply by 2. So here the osmolarity is 0.3 osmol or 300 milliosmol. So that is isoosmotic. How about tonicity? Well, the sodium chloride are non-penetrating. So the solution is high isotonic. And if you put the cell in an isotonic solution, it stays the same. Okay, how about 0.3 molar sodium chloride? If you multiply this by 2, the osmolarity is 600 milliosmol. So this is hyperosmotic. How about tonicity? Since it is more than 300 milliosmoles of non-penetrating molecules, so it is hypertonic. How about the cell? If you put the cell in such a solution, water leaves the cell and the cell would shrink. How about 0.3 glucose? Glucose is one molecule, so you don't multiply by 2. So it's 300 milliosmoles, and it is non-penetrating. That's isoosmotic and also isotonic, so the cells would stay the same. How about 0.15 molar ammonium chloride? The osmolarity is 300 milliosmoles, so that is isoosmotic. How about tonic? It is penetrating, meaning zero non-penetrating. That means the solution has less than 300 milliosmoles of non-penetrating. Since everything is penetrating, then it is hypotonic and the cell would lyse. How about urea? Urea is penetrating, so it is isoosmotic. How about tonicity? Because it is penetrating, it is hypotonic, and also the cell would lyse. Now, what determines our plasma osmolarity? The primary thing that contributes to our plasma osmolarity is sodium. Sodium is found outside of our cells. Potassium is responsible for the osmolarity inside the cell. So again, sodium is responsible for the tonicity outside the cell, but potassium is responsible for the tonicity inside the cell. Outside the cells, also in the blood, we have glucose contributing to the tonicity as well as urea, but the main thing is sodium. This means if you have high food, high salt in your food, this would increase your blood osmolarity. Okay, how does the body regulate our blood osmolarity? Of course, in case of dehydration, for example, the osmoreceptors in the hypothalamus will feel that, and as a result would release antidiuretic hormone that prevents urination. So it's going to add water to your blood, preventing urination, so the water is going to be retained in the blood, and this would help get your osmolarity down to the normal direction. Remember, adding water or taking away salt would help the concentration to decrease. So this helps the osmolarity to go down to normal. It is a negative feedback mechanism. So our receptors here are the osmoreceptors, and the center is in the hypothalamus. Then the antidiuretic hormone is the effector that gets your osmolarity back to normal. So here, if you have dehydration, your plasma osmolarity increases. You are taking away water. What would happen is that antidiuretic hormone is going to be produced and it's going to prevent urination. So water retention in the blood increases. In addition to, if you have dehydration, you will feel thirsty. And by water intake, by drinking, this will add water to your circulation, and both increasing water retention and drinking will help your osmolarity go down to the normal direction. So this is a kind of negative feedback mechanism. Okay, now some review questions. So water diffusion is osmosis. This what water diffusion is. We call that osmosis. If the membrane is not permeable to solutes, so the solutes are non-penetrating, but water can go through the membrane, then water moves from what side to what side? Always water moves after the salt. So it's gonna go from the side that has lower concentration to the side of higher concentration until the concentration is equal on both sides of the membrane. How to increase or decrease the osmolarity? Of course, uh, the osmolarity of a solution can be increased by two ways: to add solutes or take away water. To decrease the osmolarity, you need to do the opposite. So you take away water, sorry, to decrease the osmolarity, you add water or take away salt to make the solution diluted. So then you add water or take away solutes to make it more concentrated. Then you add solutes or take away water. What is the normal osmolarity of the plasma? It is 300 milliosmoles, and this is of non-penetrating molecules. The main thing that determines our blood osmolarity is sodium, because this stays outside the cell. The isotonic solution has 300 milliosmoles of non-penetrating solutes, so it resembles our plasma. If the solution is hypotonic, then it has less than 300 milliosmoles of non-penetrating solutes. Hypertonic solution has more than 300 milliosmoles of non-penetrating solutes. And how about isoosmotic? The isoosmotic has 300 milliosmoles of total concentration, regardless if it is um, penetrating or non-penetrating. Now, let's practice defining the following solutions. A solution that has 300 milliosmol of non-penetrating. How to classify this as osmotic? Here the total is 300. So this is isoosmotic. How about tonicity? Well, this has the 300 milliosmoles of non-penetrating, so it is also isotonic. And if you put the cell in such an isotonic solution, the cell would stay the same. Okay, a solution that has 300 milliosmoles of non-penetrating and 100 of penetrating. So my total is 400 milliosmoles. So as osmotic, this is hyperosmotic. About tonic. For tonic, I care only about the non-penetrating molecules, and they are 300. So this is isotonic, but it is hyperosmotic. What would happen to the cell? We look only at the tonic part, which is isotonic, so the cells would stay the same as well. Okay, a solution that has 200 milliosmoles of non-penetrating solutes and 100 milliosmoles of penetrating. My total is 300. So this is isoosmotic. How about tonic? For tonic, I look only at this part, the non-penetrating molecules, and they are 200, so they are less than 300. This means this solution is hypotonic. If you put the cell in such an hypotonic solution, what would happen? The cell would swell and lyse. Water enters the cells. Okay, a solution that has 400 milliosmoles of non-penetrating and 0 penetrating. So my total is 400. This means this is hyperosmotic and it is also hypertonic since the molecules are non-penetrating. And if you put the cell in such a solution, the cells would shrink because water leaves the cell outside to the hypertonic solution. How about if the solution has 100 milliosmoles of non-penetrating and 200 penetrating? Then the total is 300. This is isoosmotic and it has 100 of non-penetrating. So it's hypotonic and the cells would lyse. If the solution has 0 milliosmoles of non-penetrating but has 300 of penetrating, so the total is 300, so this is isosmotic. How about tonic? Well, it does not have any non-penetrating, so this is hypotonic. And if you put the cell in that hypotonic solution, the cells would lyse. Water is, of course, water has 0, so it is hypotonic and it is hypoosmotic. And then the cells, if you put the cells in water, the cells would lyse. How about normal saline? It has the same tonicity as your plasma. It has 300 milliosmoles of non-penetrating molecules, so it is isotonic. Why, when thick skin is exposed to salty water, that has high sodium levels, such as in the ocean, the skin becomes tight. Your skin cells are now immersed in hypertonic solution. Water leaves your skin cells, so your skin cells will feel dry and tight because they are surrounded with such a hypertonic solution.